Hello there, and welcome to another edition of Catalysts, a platform for the scientific community to connect, discover, and transform. This is your host, Kedar Aris, and today it's my distinct pleasure to connect with our next guest, Dr. Yasemin Sancak, Assistant Professor at the University of Washington in the Department of Pharmacology. Yasmin's primary area of research is in mitochondrial biology. Mitochondria, as some of you may know, plays a central role in various aspects of cell metabolism, from energy production to cell apoptosis. And in order to assess the cell's needs, mitochondria often interact with other cell organelles. But how these exchanges occur relative to proteins and metabolites are not well understood. Yasmin's group uses a combination of biochemistry, molecular genetics, and cell imaging techniques to characterize how these interactions affect the mitochondrial distribution and function associated with diseases such as neurodegeneration and cancer. Yasmin has an extensive publication record, including in prestigious journals such as Cell, Nature, and Science. She is also the recipient of the prestigious NIH Director's New Innovator Award, and more recently, the 2021 Pew Biomedical Scholars Award. It's my pleasure and privilege to welcome Yasmin to the show. Good morning, Yasmin. Welcome to the Catalyst podcast.
Thank you so much for having me. It's my pleasure.
Absolutely. To begin with, for the benefit of our audience, could you briefly describe your research? Why mitochondrial biology?
Absolutely. I am in love with mitochondria. They are amazing organelles, but I think my encounter with mitochondria was because of my postdoctoral lab, which a lot of fun during my postdoc. And I am in general interested in cell biology and organelle biology. And mitochondria just happens to be what I like the most.
Mm-hmm.
And in addition to that, mitochondria are involved in a lot of different diseases. Their regulation, their function is all related to neurodegeneration, mitochondrial diseases themselves. So for most of which there are no therapies. So I feel very passionate about understanding the basic biology of mitochondria so that we can try to understand diseases a little bit better and hopefully identify some targets.
Got it. I briefly looked through your PubMed record, and I was in awe because, given my background in cardiac electrophysiology, I have some exposure to the mitochondrial calcium uniporter, but not as much. And I read a recent review about some of the discoveries that have happened related to the mitochondrial calcium uniporter channel in terms of the identification of the MICU1 protein, the MICU2 protein, the essential mitochondrial calcium regulator. And some of these have been more recent discoveries. And I look up the references and they all point to your group. So it just is amazing that your group has been so instrumental in identifying and describing the structure and the function of the mitochondrial calcium uniporter. Can you briefly describe, like, some of these are some major discoveries from what I understand? How was that process like? I mean, was it inspirational? Was it incremental? It's just amazing discoveries. So.
Again, the credit goes to my postdoctoral advisor, Ramsey Musa. It was his vision to identify this mitochondrial calcium uniporter. And people knew the uniporter existed, and biochemical methods previously had failed to identify. So he came up with a essentially different method. Biochemistry didn't work, so he applied, genomics and computational biology. So he made the major discoveries, the initial identification of MCU and MICU1. And when I joined the lab, it was clear there were other components of this protein, and I just did biochemistry on that and identified one other component. Obviously, it feels great. I still remember the day I knocked down Emery and there was no calcium uptake, and then like you found something, right? So
Absolutely.
it's extremely exciting, of course, to find new biology.
Got it.
Um,
Go
yeah.
ahead. And knowing what we know now, what are some of the new exciting things are you working on in terms of our understanding of the mitochondrial calcium uniporter?
Yeah. So the biochems, the channel itself has been extensively studied since its identification. Now we have structure, we understand how they interact with each other, but there are also exciting avenues, of course, in terms of like channel biology. Because I'm not a channel biologist by training, I decided to go back to my roots and do more signaling in my own lab. So I'm mostly focusing on understanding the role of calcium signaling in regulating mitochondrial functions.
Mm-hmm.
So understanding how MCU regulates cellular signaling and mediates the communication between mitochondria and the rest of the cell. For example, we're trying to understand how MCU is involved in regulation of transcription and how that relates to some of the phenotypes that we see with loss of MCU mutations in mitochondrial calcium importer. And that's one part of the lab now. And the other component is I got very much interested in understanding mitochondrial organoid interactions. So that is something that I started in my own lab.
Mm-hmm.
We have developed a method to specifically label and isolate mitochondria that touch different organelles. And that is just, at the beginning stages, but
Right.
I'm really excited about it. So maybe we can have a biochemical handle in organelle interactions. Those are the two broad research areas in my lab right now.
That's pretty impressive. So, is this something similar to the discovery of the fluorescence proteins, which was my understanding of it, is that enabled being able to attach those proteins to describe the concentration of calcium regionally, that enabled the identification of how what the threshold is for calcium uptake of the mitochondrial calcium uniporter. And I think that was one of the key technologies that led to the discovery. Is this something similar in terms of being able to tag how the mitochondria attaches, or the physical proximity to the organelle?
No, we're not targeting mitochondria to anywhere, and it's not.
Okay, okay.
So we're just trying to look at what happens in a regular cell, and then see how certain interactions affect mitochondria. It's not necessarily focused on calcium, though.
Got it, got it. Okay. And you
A little bit more from calcium.
got it. So you're working on different second messengers other than just the calcium. Got it. That's pretty interesting. And my understanding is you primarily work with knockout mice for all your studies with mitochondria.
Yeah, I had made an MCU knockout mouse stock, and some of it is published. We haven't started doing mouse experiments yet, so just because it's a little bit time consuming, it takes longer. But that's the hope. Once we make some discoveries in a tissue culture model, which is what I would like to expand it to a mouse and understand the role of mitochondrial calcium uptake in physiology, and basically work out the mechanism in cell culture and then move to mouse to the physiology is the plan right now.
Got it, got it. And just last question before we transition from mitochondrial research. Is it fair to say that the profile of mitochondria is similar across different cell types, or is it different across different cell types?
Cell types, it's probably different. What we know for sure is that it's definitely different across tissues.
Okay.
And imagine those differences may be passed on to cell lines that come from different origins, right? But yeah, it's different tissues have different mitochondrial protein composition.
Interesting. So an adipose tissue mitochondria and the adipose tissue would be have a different functional profile than, say, mitochondria in the cardiomyocyte.
Yeah, exactly. Different proteome. Some functions are the same. Of course, like mitochondria is involved in oxphos.
Right, right.
Others are different.
Got it. That's great. And I'm pretty sure there's a lot more nuances to mitochondrial biology, and we could probably spend the rest of the day talking about it. But
If only if we knew all of it, there is still so much to discover.
absolutely. So shifting gears slightly here, I understand you became an assistant professor at the University of Washington in 2017. So it's been roughly 4 years as a PI. And I imagine it must have been a different challenge from being a postdoc, being focused primarily on research, to transition into a PI role as an independent PI, managing students, writing grants, service teaching, among host of other things. So can you kind of describe what a life of a PI as represented by you on a day-to-day basis looks like?
Yeah, absolutely. I mean, I can compare it to the life of a postdoc. I let me begin by saying that I love being a PI.
Mhm.
I was very excited when I first started. I still have the same excitement.
Mhm.
But life is very different and much busier. And as a postdoc, your main job is to do research.
Mhm.
Your time is just for research, right? You go to lab and that's what you do. But
Mhm.
PI, you have a lot of other obligations. I have to teach, I have to mentor my students, I do a lot of departmental service, university service, etc. And on
Mhm.
top of it, I do research. And, obviously I'm not at the bench as much as I used to be, but I still like to do experiments and oversee some stuff in the lab where, I do the crazy projects that are all this stuff. So life is much different, and you learn to adapt and use your time wisely, right? You can't do the same type of bench work like you did before, and also on top of it, do a lot of teaching. It's just not going to work out.
Right.
One thing that I learned, everyone learns very fast, is that you have to prioritize. You have to uh, you have to learn that perfect is the enemy of the good. And sometimes things are not perfect, but they need to get done. And in terms of busy, I'd say it's as busy as before,
Mhm.
but it's a different kind of busy. Like
Got
I had two kids during postdoc, so I only had like 8-9 hours during the workday to be in the lab, and it's the same for me. It's just those hours are being filled with different tasks.
different tasks. Got it. And how much time do you spend? What takes a major chunk of your PI life? Is it grant writing, teaching, mentoring students, or additional activities like research or establishing new collaborations potentially, or identifying new
One
ideas?
more. Yes.
You said something and Siri got activated. Do you mind repeating the question?
Yes, absolutely. So my question was, in terms of you getting pulled in different directions, teaching, service, mentoring, grant writing, doing research, which takes the biggest chunk of your time right now?
I personally prioritize mentoring my students. So, by default, I think I spend most of my time on it.
Got it.
Everything else comes second. And, like mentoring students also means being into our research, so I may just like same category. And in terms of grant writing, teaching, departmental service, they are very seasonal in a way.
Follow the cycles.
Yes, exactly. So most of my times I think like research slash mentoring students, I think, which is great because this is what I want. But like there was a time last year where I was starting to teach a lot of classes, and I spend more time on teaching than other things.
Got it. And did the pandemic impact your lab research? I imagine it did. And if so, how have you been able to navigate that pause, if you will?
Yeah, it did impact significantly. I mean, many labs were shut down for months.
Mhm.
It's not just, when you shut down for 4 months, you just don't lose 4 months. You lose the momentum, and
Yeah.
like you had undergraduates who were doing great, they couldn't come, and then they graduated, so their research stopped, etc.
Mhm.
Navigation, I mean, there isn't much you can do, you know.
Yeah,
Um,
it's a once in a century pandemic. Nobody knows how to prepare for it.
exactly. We try to, as a lab, meet every week, do more reading, do more writing as much as we could. And we did work in shifts.
Mhm.
Everything was slower. To navigate, we're just back in lab and like trying to go, be as productive as possible now. But loss is loss, what can you do about it?
Right, right, right.
I guess the best thing you can do about it is being zen about it and just like don't dwell on it.
That's great advice. Uh, Speaking of DP2, the grant that you received, I mean, it's such an amazing and prestigious grant. I mean, obviously there are R01s and R21s, and but I've very seen very few people actually win the DP2 awards across my professional network. Uh, so I was wondering, from your point of view, how similar, how different is the grant writing for a DP2 compared to, say, an R01 or an R21?
It's pretty different, and the spirit of the grant is different, right?
Mhm.
You have to have that idea that no one has tried before. When
Mhm.
it works, it's going to unleash this amazing biology that no one could answer before.
Mhm.
As opposed to an oral one, which is more, still very exciting biology, but you have to show that it's going to work, basically.
Mhm.
So it is very different, and the format is different. There you don't need as much preliminary data, and it's a little bit shorter. And I can't remember. You could like there's no codified structure basically, so it's more fluid in terms of its structure. I think as long as it's a good idea, as long as you are the person to do it, um, it may work.
Got it. And so, if preliminary data isn't as much of a focus, my sense is storytelling would be a more important focus in terms of being able to tell a good story of what that idea could do.
Um, yeah, exactly. I mean, it has to be solid, of course. And I did have data showing like strong feasibility.
Oh, you did. Okay. And you included that as part of your, okay.
Required, but I guess it doesn't hurt to have.
Absolutely. It doesn't.
And yeah, absolutely, it has to be like the idea has to be scientifically
Compelling. Yep.
and feasible, but no preliminary data required, as far as I know.
Got it. And was it, were you successful the first attempt, or did you have to submit a revised application? Okay.
Yeah, I revised it based on the first time around. I didn't get it, and
Okay.
the reviews were very helpful. And I revised it based on the reviews and rewrote sections. So getting feedback is always good, of course.
Absolutely.
Got it the second time around.
And that effect, and this, in my understanding, and I might be mistaken, this is primarily for early career scientists, so it affects your ESI status now. So
I don't have that sweet ESI status anymore.
more after the DP2, well, that's what it's meant for. Like, once you get your DP2, that's a R01 effectively. Got it.
Exactly.
Okay. uh, the next area I wanted to kind of briefly inquire was your recruiting process to a faculty position from a postdoc. And obviously, you are at University of Washington, which is an amazing research university. So my question was, what attracted you to University of Washington? What was this criteria that effectively said this is a good place for me?
Good question. So, everybody's circumstances are unique. And for me, we had a two-body problem.
Mhm.
And University of Washington offered the best solution to our two-body problem. That was a major determining factor, to be honest.
Mhm.
But this said, obviously, we wouldn't have come here if we didn't feel like this was a very supportive environment and we had everything we needed to succeed and do our research. So, I mean, beyond that, I think things it doesn't matter that much. And I knew I was going to have access to good students, equipment, space, mentoring. Beyond that, it's yeah, it's splitting hairs. I think
Got it. So,
that was the major.
so primary motivation was the two-body problem and the solution to that two-body problem, and University of Washington effectively provided a good solution to that.
He's in the same department. He has his own lab, and he's doing great too. So it worked.
Got it. In terms of navigating the whole interview process, uh, I understand there's a public talk and there's a chalk talk where they ask you about, okay, what's the planned future in terms of future R01s or whatnot. And you can prepare for some of it, but how much of a mentoring is required to kind of come up with your own spreadsheet for startup package or negotiating salary or whatnot? And oftentimes that's not something that a postdoc is ever trained in. So how did you navigate that process?
Yeah, I think you're right that we're not trained in doing most of the stuff, but it really helps to talk to people who have gone through this recently and learn what to expect, such as like you're doing now.
Mhm.
And yeah, there is the public talk component,
Mm.
but a job talk is different than any research talk. So
Right.
I really recommend everyone to practice, and get feedback from others. And hopefully there are people, both senior and junior, who will be willing to listen to your talk several times, different iterations, and give you honest feedback.
Mhm.
Tell you that you should change this and that type of stuff. That really helps. And um, that's one thing how you can prepare. There's also a lot of information online.
Mhm.
In terms of like, I think the biggest black box is a little bit the chalk talk.
Mhm.
I would say there is not a single format for it. And I will highly recommend, like if you get to that stage, reaching out to the person who's your contact person and asking, what are you looking for in a chalk talk, so that you don't go there with slides and they're like, no, we can't have slides. Like what they want and they'll be willing to share. Why wouldn't they, right?
Right. Right.
Um, some places want to know what the three aims of your R01 are.
Mhm.
Some places say, we don't want to talk about your R01. We want to see what you're thinking, ahead. Like, what do you want to do 10 years from now? Tell us your crazy ideas. So without asking, you're not going to know. But so I think it's important to like talk with the search committee at that stage.
And they would be happy to share information because it makes for a better conversation.
Yeah, absolutely.
Got it. One of the things I recently found out as I was talking to my PIs and other mentors was the idea of fit with the department. And my sense always was, how does your research fit with how the rest of the faculty in that department does research? And I used to initially believe that as long as it's complementary, you're a good fit. But what I'm slowly beginning to realize, and I could be completely mistaken, and I'd like to hear your thoughts on that, is if you're doing something which somebody else in that department is doing, you're automatically rejected. It's like you got to bring something to the table that nobody else does in the department, but at the same time have opportunities to collaborate. So it almost seems like a delicate balance. You cannot be too far off where there's no fit, but you cannot be too close where you're overlapping with what some of the other people are doing, which then makes you redundant or your research redundant.
Yeah, I think I agree with you. I think there is probably a delicate balance, but I don't think every department or search is for one particular person. I know, for example, my department during the search, they said, let's just bring the best people in, no matter what the research are, because we want to diversify anyways. So, and we want the best people that we can get. And that's one way of doing it, right? And University of Washington is so big that it's impossible, not to have other mitochondrial biologists, and they're not
Right.
just not have me just because I do mitochondrial biology, right?
Right.
But other places may be a little bit more picky, and I would say like probably if they had another person working exclusively on mitochondrial calcium uptake, it probably may not be a good fit, right?
Um,
But, yeah, I think it may differ from place to place, department to department, and but you as like if you're offered, if you're I think of what is it,
got it.
support or collaborators or diversity. If I want to do something that I don't have expertise for, can I easily find it in this institution or nearby institutions, etc. So yeah, fit is important, but not the you know it's there's no one formula basically.
Got it. Got it. So as you look forward 5 years from now, what are some of the challenges that you see coming down the pipeline for you, either in terms of research or in terms of uh, the grants or recruiting students, or just in terms of things that everyone should prepare for as a PI?
In this job, publications and money is the current, grants are the currency.
Mm,
I guess they are the most challenging for everyone.
right.
So, but I think if you are doing good science, something you're proud of, the rest will follow. You'll get publications, you'll get grants, etc. So, like, I don't know what type of challenges to predict, honestly. Um, maybe NIH budgets maybe will be.
So I hear they're expanding it by 20%, hopefully. So,
I recently expanded, so getting more grants will not be a challenge. Um, yeah, I think yeah, I can't honestly predict this or that necessarily.
mhm.
We'll see.
And in terms of, I mean, I understand you finished your undergrad in 2002 at Boyadici University in Turkey, and then it's almost been 2 decades uh,
Mhm.
to where you are right now. Looking back, have there been any inflection points where you wish you had done things differently, or you were glad you did it the way it did, so that it eventually ended up getting you to where you are as a successful PI? Uh, maybe you could share some of those experiences, perhaps.
So, good question. Um, I personally don't regret anything. Um, I may say maybe if I had kids a little bit earlier, that will make my life a little bit better now. Because starting a career with young kids is a little bit harder than starting a career with maybe older kids.
That's true.
I would say that, but otherwise, it has been a lot of fun. I
Mhm.
PhD and postdoc a lot, and I'm still having fun as a young faculty. um, I try to be very methodical and thoughtful in every process, and um, maybe it's boring, but you want to say, what happens if I do this? What happens if I do this? And pick the best option, and then also, you know, be zen about it a little bit. Like you can't control everything. You
Right.
just do your best and hope that it works out. Sometimes there's luck involved in this in life, not in
Right.
just but in life. Um, yeah, I that's that's my advice. I'm thinking, would I have done anything differently? I think so. Yeah.
You shared two great advice. Make sure you have fun, because this research or the career of an academia is high stress environment, and if you're not having fun, I don't know what's the value in doing it. So the fact that you suggest that make sure that you have fun is a key advice that anyone listening to this should definitely pick up. And the second thing, and I agree with you, is being zen about it, because there are going to be so many failures in terms of writing grants and getting rejected and so on and so forth, and failed experiments and whatnot, that if everything gets taken personally, there's going to be a lot of heartache and pain. So,
Especially for the job search, it's
mhm,
hard. You have to have a nerves of steel.
mhm,
You are being evaluated from, your science is being evaluated and criticized. You as a person, it can be traumatizing.
right, I bet.
At the end of the day, you just have to say, okay, start looking for a job, saying that I know I'm not going to get every single job that I apply to.
Mhm,
After every interview, I think, just um, enjoy the process. You're making new connections, you're seeing new places,
right.
maybe do your best. And if you didn't get the job, look back and say, can I learn? Yeah, did I do anything wrong? Could I fix something, etc.
Got it.
And sometimes you do a great job. It's just, they decided it's not a good fit. It happens.
Yep, not all great ideas get funded. Not all great people get the first job that they apply for. So it's part of the process. Great advice. I know I've taken a lot of your time, so I wanted to take this opportunity again to thank you so much for coming on to the Catalyst podcast and sharing your thoughts. We greatly appreciate it. With that, we will wrap up this episode of the podcast. If you liked what you have heard, I encourage you to share this episode with a friend. I'd also love to hear from you and how you are striving to be a catalyst. So I invite you to leave a comment on my Twitter or website. This is your host, Kedar Aris, signing off. Thank you for listening, and until next time, connect, discover, transform.